The overall goal of the following experiment is to study the extent of dendritic arborization in the hippocampus of a mouse model. This is achieved through two different methods. In the first method, dendrites are traced manually in real time throughout the entire thickness of each section. Following tracing, the entire dendritic tree can be reconstructed and analyzed using the fan diagram. Analysis of various fan diagrams derived from different mice can be used as an objective means of comparison. The second method is to visualize dendritic arborization with extended depth of field imaging. Finally, a panoramic method is used to stitch multiple high-magnification images together to yield one high-resolution composite image for qualitative and quantitative assessment of dendrites in the entire region of interest.
Demonstrating this procedure will be Mojabi and Ponnusamy research fellows from my laboratory.
The main advantage of these techniques over existing methods is the ease of use and speed of acquisition and data collection.
This method can help answer key questions in the field of neurobiology, such as assessment of dendritic arborization in affected brain regions and evaluation of the effects of different therapies on dendrites.
In this procedure, one day after the mouse brain has been fixed in 4% paraformaldehyde, place it in sucrose solution for dehydration for 48 hours at 4 degrees Celsius. Remove the brains from sucrose.